The last MAPS presentation of this year. As you will see on a slide in a couple minutes, we will resume in January with a great program that Jennifer McDonald, Chair of MAPS, and she will introduce the speakers in a couple of minutes. As you see up on the slide, we have the members of the MAPS committee. If you ever get a chance, thank them. They do this voluntarily. There's a lot of work involved, and they've set up another great schedule this semester and next semester, and probably from now on. How many have visited the Great Valley Museum? Well, the rest of you should take advantage of it. It's one of the best places for families to visit in the area. It's a learning experience and some beautiful dioramas there. I strongly recommend you join as a family, because the membership will get you into many museums around the country. I know I've often saved a lot more money than the cost of the membership. I think it's about $140, but you'll get into a lot of things, even in San Francisco. On the first Friday of every month, the Great Valley Museum and several of the faculty hold a science night at the next door. And the telescopes are usually available if it's a clear night for viewing. And these wonderful faculty will help you, give you a tour through the sky. It takes some funds to run the program, and we especially want to thank the City of Modesto and the Stanislaus County Office of Education for their donations, as well as these other organizations. Modesto JC, in addition to the MAPS program and the Science Colloquium, also hosts the annual Regional Science Olympiad. It'll be on this campus on March 14th. Come on over. It's fun to watch the junior high and high school kids compete. A lot of them are very good for spectators, and you'll enjoy and cheer on our future scientists. In addition to MAPS, we also have the Science Colloquium. There's one more this semester. It's an interesting one on biomass and biomanufacturing. The MESA program at MJC will be very involved in giving that talk and the status of what they're doing. The next MAPS talk is on January 23rd. University of California, Merced, Emily Lynn Williams. From water woes to smoky air, I'm just reading it, but it's on climate change. This is one of the biggest challenges we face in our society. We've got to start doing a lot more than we've been doing, or we're in big trouble. So turn out and see what she has to say. And the rest of the semester's up there, February 27th, and and April 17th are in the Student Center instead of in here, and they're family events. That is, they're for people of all ages. We've had the first group here before twice. They put on a great show from Foothill College. And the one on April 17th, I don't know that group, but they will be doing a bunch of demonstrations. It'll be a lot of fun, and it's good for all ages. Adults can learn just as much about science at these shows as the kids. And finally, we have our talk tonight, and our chair, Jennifer McDonald, will introduce the speakers.
Okay. Well, thank you all so much for joining us this evening. And if you are going to be looking for something to sign in, you're going to come see me down front after the talk over here in the corner. So without any further ado, I would like to introduce Dr. Kyle Fink and Dr. Julian Halmai, and they are both going to come tonight to share with us what CRISPR is, this amazing technology that has truly revolutionized how we can interact with cells and treatments that are now possible that were never before possible. So I'm going to go ahead and step out of the way and turn this over to you. Would you like a microphone?
Awesome. Thank you so so much for the kind introduction. Again, thank you to the community. Thank you for the organizer for inviting Dr. Halmai and I to come and share a little bit, I think, of what we do. And so we're located, both Julian and I, we're located at the UC Davis School of Medicine, so our labs are located in Sacramento. We're both members of what's called the Institute for Regenerative Cures as part of our Stem Cell and Gene Therapy Center. And so a lot of what we're going to talk about today is work that's currently ongoing in our labs, and we're going to give you a little bit of background on, kind of CRISPR, and I'm going to give a little bit of background of maybe where we are as we think about therapeutics, how we start to develop treatments for really what we consider rare and severe unmet medical needs, and then I'll turn it over to Julian to kind of maybe tell you about where we think the future of things are going and some of the work that's been coming out of both of our labs. Okay. And so the first thing, and a lot of times when I start a talk, we like to frame it around the idea of why are we doing what we're doing. And so a lot of what you're going to hear today is going to be focusing on rare and ultra-rare disorders. And again, this comes from the National Human Genome Research Institute, and it originally from somebody at the NIH, Tim Miller, who put this slide together. And so collectively, when we think about rare diseases, they're not rare. And so approximately 1 in 10 Americans, or 30 million in the US, or 400 million in the world, are suffering from a rare disease. And so when we think about rare diseases, this is going to impact more people than AIDS and cancer combined. And so when we really think about cancer is a big, big problem, rare disease is a big problem. Even more so, about 50% of those that are affected by rare disease tend to be children. And then out of these, about 80% of them are going to have some form of genetic cause or genetic underpinnings. And that really starts to become important as we start to talk a little bit more about kind of current technologies, things like CRISPR, gene therapy, and how we can start to do these targeted interventions. And then even more so, when we think about rare disease, is right now 95% of these, of the rare diseases, don't have an approved therapeutic. There's no FDA compound, there's no treatment. We hear stories a lot. We interact with the rare disease communities quite frequently. We go to family days, we go to education days where we're interacting with those that are suffering from these disorders. And the heartbreaking thing that we hear more and more often is go home and love your child. Go home and there's nothing there. Maybe some research will come someday. And so again, not only is this affecting a very large portion of population, it tends to affect children, and there's a large unmet medical need. So again, hopefully I'm convincing you that studying rare disease is not rare, and it's really important to do. But we're also living in an incredibly exciting time in terms of treatments and as we think about rare disease. And so when I started in science, not that long ago, because I'm not that super old, but I'm getting older in science, is we used to talk about, well, it's going to be great. Someday we'll be able to treat rare monogenetic disorders. Someday we'll be able to do this. Someday we'll be able to restore. If you have a genetic mutation, could you replace it? Could you repair it, right? And this was always kind of a pipe dream. And so kind of in my lifetime as a science, gene therapy is now a thing. And so when I started in science, we said, well, gene therapy, you have a genetic mutation, could you just put the gene back that's missing? Again, this was a lot of theory when we first got started, but now there's 7 approved FDA compounds that if you happen to have a form of genetic blindness called LCA, spinal muscular atrophy, which is floppy baby syndrome that you may know of, some forms of hemophilia, Duchenne's muscular dystrophy. Now you can actually go, if you had a child with a mutation of SMN1 and your child had SMA type 1, you could get a prescription written for you for Zolgensma. The story of Zolgensma is with spinal muscular atrophy type 1, these children typically don't exceed their 2nd birthday. So they have a lifespan of about 24 months, can never sit unassisted, can never lift their head. And again, it's a very, very severe disorder. Now, if they receive intravenous injection of AAV9 containing the gene SMN1, these children are now living into their 2nd decade of life. And if they're treated early enough, they're actually almost indistinguishable from those that don't have it, right? So again, gene therapy is something that is incredibly impactful, and it can be life-saving, and it can be curative. This is really great, and I think in 2017, 2018, 2019, we all said, well, this is great. We're just going to go down the list of every single monogenetic disorder, and we're going to make a gene therapy for it, and we're going to cure them all. As we'll hear as the talk goes on, this is a lot more complex, and there's a lot of nuances. SMA kind of tended to be the poster child of why this could be so successful, and then also with forms of congenital blindness. A couple that we're really interested now and are having positive results are in something called Rett syndrome, and so there's currently clinical trials that are ongoing. Industry is still kind of interested in this, although the interest is somewhat waning. And again, these are in girls that have mutations in a gene called MECP2 that results in kind of normal development for the first year to 3 years of life, and then they undergo this very severe regression where they lose the ability to walk, they lose the ability to talk, and they have a lot of seizures. And now we're showing that even in people that are 21, 22 years old, is they're starting to regain some of these functions after they're receiving a gene therapy. And so again, we're kind of living now that gene therapy is a thing. Gene therapies are prescribed. They're part of the FDA. Insurance will reimburse some of it. On that previous slide, we had costs, but we don't have to talk about that right now. We'll save that for later. And then also, I think we're also living in the dawn of CRISPR therapy is now a thing. And so not only could we go in and replace the gene that may be missing, in Casgevy is now an approved FDA compound that if you have sickle cell disorder, you could get a CRISPR therapeutic prescribed to you. Again, this isn't in vivo gene editing. This is, we're going to take out your blood progenitor cells. We're going to knock out a gene that represses the expression of fetal hemoglobin. But then we could deliver those cells back to you, and your body will now express the appropriate amount of hemoglobin and functionally cure your sickle cell. This costs about $4.1 million. Only a handful of patients have actually been treated with it. It's very long, it's very expensive, it's a very challenging process. But now there's people living today that are CRISPR corrected. They've had their genetic mutation, or they've had a genetic targeted double-stranded break made, and now people are living with CRISPR-corrected cells in their body. Except this is ex vivo. And so one of the things that I think has captured a lot of excitement and public and everything else was recently, Julian and I were at the American Society of Cell and Gene Therapy, or ASGCT, and we got to listen to an amazing talk by Dr. Kieran Musunuru describing the first in vivo base editing correction in somebody named Kyle Jr. baby KJ. And what they actually were able to do is identify a specific mutation in a gene that's very critical for the urea cycle, that if you have this mutation, this loss of function mutation, or you have this mutation in your genetic sequence, is that when you eat protein is you're going to start to accumulate a toxic level of ammonia in your blood that eventually results in severe neurotoxicity. Patients typically have to go on extreme protein deprivation diets very early on in infancy, and eventually will require a liver transplant. And so when this child was born and was noticed very quickly that they had this urea cycle disorder, on day 2 of life, he became ill and showed this rapid spike of ammonia levels in their blood. They genetically sequenced him and found that he had this biallelic CPS1 variant, which is the most severe type of this urea cycle disorder. And again, the standard of care for this is liver transplant, and again, whether or not they could find a donor or not. Again, why this has been so public and so renowned is that baby KJ was actually able to receive a bespoke targeted therapeutic to correct his mutation using CRISPR base editing technology delivered with a lipid nanoparticle. And they were able to do this within 8 months. And so before baby KJ turned 8 months of age, they actually had delivered multiple doses to him. And so again, when we see this and we think about the nimbleness, the tools, and the development of CRISPR, again, these can be life-saving. Baby KJ is still living, he's at home, he hasn't required a liver transplant, he's still on a normal protein diet. But a lot of times we talk about how this could be done within 8 months. And really what this is years and years and years of really hard work to develop the tools, to develop the technologies, to first be able to find the correct variants of CRISPR-Cas9 that can do base editing, different types of base editors, to screen these in cell models, to knock in genes that represent the different mutations to show that you could get the correct amount of corrected mutations with minimal bystander or indels or off-target mutations, to test this in transgenic mouse models where you just put in the sequence and you screen this and you look at off-targets, eventually to this massive undertaking with biotech and pharma to develop a therapeutic and a lipid nanoparticle that they could dose him. And so here you can see in the upper left the protein intake. He was on a severe protein deprivation diet. The dotted line is normal protein intake. You can also see the ammonia levels over on the upper right. I'm going to put the laser pointer on here so I can point to it. And again, what we can see is these spikes of ammonia levels to near toxic levels that would result in neurotoxicity. But following the 3 doses of this base editor delivered by a lipid nanoparticle, is that now, corresponding with an increase into a normal protein diet, that baby KJ is still having non-toxic accumulation of ammonia. And this is him that is now living at home as one of the first humans to ever have an in vivo CRISPR correction delivered to him that's theoretically curative. He hasn't required a liver transplant yet. So again, I just kind of wanted to set a little bit of the groundwork. We're living now in the age of gene therapies. We're living now in the era of in vivo CRISPR editing. And what I want to do now is turn it over to my colleague to go through a little bit more about what we think about in terms of CRISPR tools and kind of the next generation and how we could implement those for these rare genetic disorders.
Thank you so much, Kyle, and thank you all for spending your Friday evening with us. I really appreciate it. And also, again, thanks Jennifer and the committee for inviting us. So, as Kyle already introduced, specifically from the perspective of rare diseases, there's a huge unmet medical need. So out of the 20,000 genes that we have in the human body, approximately 3,000 or so can be considered part of what we would call the druggable genome. These usually fall into 3 different categories, including G protein coupled receptors, ion channels, and kinases. These are proteins that have some kind of active pocket that a small molecule drug can easily bind to and elicit an effect. But in reality, the existing pharmacopoeia, so the actual availability of small molecule drugs, is only a few hundred targets. So that begs the question, how can we make the genome druggable? And as Kyle already introduced you guys to, and as most of the reason why you're here, CRISPR maybe allows us now to actually target a lot of these genes. But before we talk about the druggable genome, let's actually talk about the disease genome, just so everybody is on the same page here. So normally you would inherit an allele from your father and from your mother. And sometimes what occurs is that mutations can be inherited from either parent, or they can actually happen de novo. So that means they happen somewhere along the lines of embryogenesis or before in the germ cells. What can go wrong now is, for instance, a person can only inherit one functional copy of a gene that might not produce enough protein to actually have a normal function of the cell. That will be called a haploinsufficiency. There's about 600 genes or so that are estimated to be causing haploinsufficiency due to this one allele present. Likewise, there's also recessive disorders, so that's the situation where you inherit an allele from both parents that is defective, and there's about 500 genes that cause disease in a recessive manner. And really, in this case, a person has no functional copy of the gene. Another example of sort of this quasi-haploinsufficiency or loss of function in a recessive fashion or in a dominant fashion are actually X-linked disorders. So this is an example of a male, so where there's only one X chromosome present, which is the traditional case of X-linked disorders. The X chromosome, as you'll hear about a little bit later, is the hotspot for intellectual disability genes. There's actually about 140 or so genes that are involved in cognition and neurodevelopment. And then the last category are dominant disorders. So about 65 genes cause disease in a dominant fashion, so where there's only a single mutated copy that actually causes disease. As Kyle already introduced, the idea of gene therapy in this very elaborate cartoon here is we package a coding sequence of a gene into a viral vector. So this will be an adeno-associated virus, which is considered to be the gold standard because it's somewhat safe and actually can get delivered pretty well to an individual, either through bloodstream or through direct injections in the target tissues that you care about, that then goes to the cells where it's needed and elicits an effect. Now, how does this work in terms of the druggable genome? So I already introduced the idea of the disease genome. Gene therapy would be fantastic in cases where haploinsufficiency is happening, where you don't have enough expression, and in a similar fashion where you have a recessive condition, where the idea again is you just don't have enough messenger RNA, so the gene therapy would replenish that messenger RNA. But AAV comes with some challenges, and the biggest one is actually the size limitation. So an adeno-associated virus can only carry about 4.8 kilobases of genetic material, and just taking the 660 haploinsufficient genes, about 135 of those could, for instance, not get packaged into an AAV because these would be too big to package. And this is what you can see in this figure here. This is a plot that actually shows that as soon as you go above these 4.8 kilobases, the AAV gets refragmented, the AAV doesn't package very well, and from a clinical perspective, from the translation of these tools, this would not be something that would not pass. Another example are actually dosage-sensitive genes. So one of the other challenges with AAV is getting to the right level. So we would call these dosage-sensitive genes also Goldilocks genes. And a really good example of that is the X-linked gene MECP2. When you have too little MECP2, this causes Rett syndrome. So this is an X-linked disorder in females that have a defective copy of that would represent a 0.5x MECP2 expression. If you only have one functional allele in the mosaic brain, that would cause disease. Likewise, if you actually have a duplication of MECP2, it also causes disease. That would be called MECP2 duplication syndrome. So you really have to hit that sweet spot of expression. And it's really, really challenging to do in a complex system like the brain when you deliver a coding sequence of a gene that is driven by a really strong promoter. The gene therapy that Kyle introduced earlier for Rett syndrome actually has some nifty tricks incorporated already to control for that expression, but it is something that took a lot of time to develop and actually is maybe also unique to certain genes and not applicable to others. So what that results in is that there is a need for physiological gene regulation. And then the other extreme, this seems maybe somewhat obvious, but if we think about a dominant disorder where there's a gain of function, so the protein that is mutated has a completely new role. So for instance, it's topically expressed, so it has new binding partners that it shouldn't have. Expressing more of a wild-type allele in this case wouldn't fix the situation because this would just like to get engulfed by this mutant protein that's already present. So that's why I want to introduce real quick the idea of epigenetic gene regulation. So every gene in the human body, in any organism that actually has epigenetic control, has this spatiotemporal regulation of expression. The spatiotemporal regulation of expression is mediated through the activity of specific activators or repressors. So for the repressors, they would put a gene state into the silent state, which you can see on the right here, through different positive feedback loops, as well as transcriptional activators that can actually overactivate a gene into an active state. So you can think of, for instance, a gene that should only be on in a neuron gets pushed to this state versus a liver gene in that neuron should get pushed through these epigenetic regulations into the silent state. There's specific layers that contribute to epigenetics. One of them is DNA methylation. So DNA methylation is probably the most studied epigenetic mark. What can happen is that DNA gets methylated in the cytosine residue in a CG diet. And what this is associated with, to some degree, is when you actually have a promoter region of a gene. So this is where the gene transcription starts. If this gene promoter is hypermethylated, this gene gets silenced. So this is often something that we observe, for instance, in cancer for tumor suppressor genes, where these tumor suppressor genes become inactivated through DNA hypermethylation. Another layer that most of you are probably very familiar with is the idea of DNA has evolved some very sophisticated mechanisms to fit about 6 feet of DNA into the tiny nucleus of a cell. The way the DNA does it is it wraps around nucleosomes. Nucleosomes are made out of these core histone proteins, H2A, H2B, H3, and H4. And these histones actually have tails that are protruding from them. So these are amino acid tails, and these tails can become post-translationally modified. In reality, there's a whole bunch of modifications that you might be able to see here, and they all listed different effects on gene expression. So this all gets combined into some kind of model in which we can envision an open gene promoter be depleted in DNA methylation and having active histone marks, while perhaps an inactive gene or a gene that is sort of on the verge of becoming active or staying inactive, what we call bivalent or poised, has perhaps a mixture of both active and repressive histone marks. And then there's also the opposite, where we actually have heterochromatin. So this is really the tightly wound DNA that is inaccessible to RNA polymerase, that is unable to actually make mRNA. We also have a pretty good idea of what all these writers and erasers are that play a role in depositing these epigenetic marks. So there's different complexes that, for instance, deposit DNA methylation. So there's enzymes for that. There's enzymes that can actually deposit acetyl groups onto histone tails. There's enzymes that can remove these acetyl tails, and so on. There's also enzymes that can then read specific marks, and they all interact with each other and play a critical role in this epigenetic regulation of gene expression. Now, what does any of that have to do with CRISPR? I am really, really fascinated by epigenetic mechanisms, and I'm equally fascinated by the ability to customize and target genes in a very cheap and very democratized way too, where actually you can very easily design a guide RNA for a CRISPR targeting strategy right now, and you can order one for $3 about, if you have an online account through university, I guess, and order it to your lab, and then you can say, hey, I'm really interested in studying this specific gene, and do some kind of modification with it. So the way Kyle introduced CRISPR earlier was as a base editor or a DNA scissor, but the idea of gene regulation is not necessarily new. There have been multiple generations, so the first generation were actually meganucleases. These are similar to restriction enzymes that you use in the lab that recognize a specific DNA sequence always at the same DNA sequence. That doesn't make it very customizable, but still allows you to actually cut up DNA and do perhaps specific integrations. This has been evolved by using zinc finger proteins first. So zinc fingers are actually mammalian occurring or eukaryotic transcription factor motifs that you can actually tie together to recognize specific DNA sequences. Then there's the forgotten middle child. So these are TALs, the transcription activator-like effectors that were really quickly surpassed by CRISPR technology. What you can see in this graph here actually is we're using CRISPR in a slightly different way. So this is the next generation beyond the cutting approach, but we're using a nuclease deficient Cas9. So that will be called a dead Cas9 or a catalytically inactive Cas9 that can still bind to the DNA when it's targeted with a guide RNA, but no longer elicit a double-stranded break. Now, the whole reason why I introduced these writers erasers and all these epigenetic concepts to it is the effector domain can be one of these enzymes that are involved in epigenetic processes. So what you can see in this graph here is that theoretically, if you have an active gene that you don't want to be active, you can perhaps target specific writers and erasers to the active gene promoter and turn it into an inactive state. And vice versa, if you have an inactive gene, you can target specific domains via your CRISPR molecule and activate that gene. One really good example for such activation strategies is CRISPR activation, in which actually we would fuse an activator domain to Cas9, to dCas9, which is this viral protein derived, so it's a herpes simplex virus derived protein called VP16 that you can actually fuse multiple VP16 copies back to back to turn it, for instance, into a VP64 if it's a tetramer. That can then, when targeted to a haploinsufficient efficient gene, upregulate messenger RNA expression. So that's a sophisticated way now for us to go in and target these genes, for instance, that wouldn't be able to be targeted with an AAV because of size limitations. By just overactivating the expression of the wild-type copy, this might also overactivate the mutant copy, but the idea is the mutant copy is inactive anyways, so what does it matter? And the major advantage here compared to regular CRISPR technology is now you no longer have to go mutation by mutation, but rather you target a shared DNA sequence that should be present in every patient, which is the promoter sequence, and all you would do is drive expression of that allele regardless of what the mutation is. So you wouldn't have to develop a base editor for 20 different mutations causing FOXG1 syndrome, for instance. Which leads me to one of the examples that we studied in the lab. So FOXG1 syndrome is a haploinsufficiency. It causes severe developmental delays when there's mutations in FOXG1 syndrome. It causes also a early onset hyperkinetic movement disorder. Many individuals have epilepsy, suffering from FOXG1 syndrome, and sometimes you also observe things such as structural anomalies where, for instance, the corpus callosum doesn't develop, so that is a part of the brain that links the hemispheres. Other comorbidities include hypotonia and other issues including visual impairments and microcephaly, so a small brain. The way we have tackled FOXG1 syndrome is by now designing such a CRISPR activation strategy where the goal was to upregulate FOXG1 expression, or actually first build a tool that can do it in a clinically relevant way. And this is what you can see on this graph here on the bottom left. We developed this modular system in which we broke up Cas9 into two halves. So we have an N-terminal half and a C-terminal half of this dCas9 protein. The nice thing about this is it allows you to kind of mix and match specific activation domains that are either fused to the N-terminus of Cas9 or fused to the C-terminus of Cas9. And the goal here was to actually say when we fuse weak activators or stronger activators and kind of multiplex or synergize their activity, can we actually drive FOXG1 expression up perhaps have a effect on the cells that we tested this in that would correspond to some sort of genetic rescue. And what you can see here in this in our recent manuscript is that when we target FOXG1, the FOXG1 promoter specifically with all these activation domains, we can actually significantly increase protein levels. So this is a Western blot where we just probe specifically for FOXG1 protein. That's the top band. The bottom band is a loading control that you always need as a control to make sure that you don't actually put twice as much protein in the samples that look really good. And this is just proof of principle to show, look, we can actually upregulate FOXG1 expression, increase expression. Then we went ahead and actually also tested this in different disease models of FOXG1 syndrome. Specifically, what we have access to are patient iPSC lines. So iPSC lines are cell lines that have been reprogrammed often from skin biopsies or from blood to be turned into some kind of stem cell. So you don't need to actually extract any kind of embryonic stem cell. You can actually do it from somatic tissue and create these iPSC lines directly carrying the patient material. So these are iPSC lines that have a patient mutation that we can then also actually go in and CRISPR correct. So what you can see on the bottom graph is either this W308X mutation, so this is a specific stop codon mutation, as well as an isogenic control. So this is a CRISPR-corrected control in which we replace the mutant copy, the mutant gene, or the mutant allele, I guess, with a corrected base pair that then serves as a baseline. When we do our activation strategy in these cell lines, we actually get about 50% to 60% or so expression of what the wild-type allele, of what the corrected cell line is. What we don't know yet quite with this is how much is actually necessary to rescue. So there could be as little as maybe 10% rescue that is necessary to increase and maybe make such a syndrome better, but perhaps you actually need to get to the full 100% expression of what you would observe in the wild-type allele. We are applying this as a platform technology to, I want to say, 6 or 7 different, maybe more haploinsufficiency disorders, where we're actually testing this one by one for other conditions, and we're trying to understand how well do other haploinsufficient genes respond to such a strategy, with the also outcome of what is the actual functional rescue underlying these approaches. But in the interest of time, I want to move on to another example in the lab, which are actually, this is really where my lab is really interested in, are X-linked disorders. So in female somatic cells, so in any somatic cell of a female individual, there are 2 X chromosomes. In these female somatic cells, they start expressing, one of the X chromosomes starts expressing this long non-coding RNA called exist that causes silencing of one of the 2 X chromosomes. So the idea here is, similar to what I introduced with Rett syndrome, that we need to have gene dosage set at 1 for many of these genes. We don't want to have twice the expression that a male has in females, for most genes. And this is again mediated through the expression of this long non-coding RNA called exist, that is actually able to remodel epigenetics. So what it does on the inactive X chromosome is it really pushes genes on the inactive X into the silenced status via DNA methylation and other epigenetic hypercompaction. And this is then maintained throughout the life of that somatic cell. Now, 15% of all known intellectual disabilities are actually found on the X chromosome. So again, this is really a hotspot for brain development. As a member of the neurology department at UC Davis, this is a very hot topic for us, and obviously a very important area to target, now applying these similar CRISPR strategies. One example that Kyle's lab and my lab are both studying is the CDKL5 deficiency disorder, which is one of these rare X-linked intellectual disabilities. So mutations in the CDKL5 gene, which encodes for a kinase, causes an early onset intractable seizure syndrome that is also again characterized by intellectual disability and other developmental delays. There's also motor issues, so many individuals have have limited mobility, impaired fine motor skills. They also have hypotonia, so low muscle tone, as well as stereotypies. So that means stereotypies are just repetitive hand movements or repetitive movements in general. They also have cortical visual impairments, so they have eye visual problems, and other comorbidities. So when you talk to parents, actually, what would be your favorite thing to improve in a child for the quality of life, it often comes down to sleep problems as well as feeding difficulties. And then obviously also the seizures, because nobody wants to see their child have a seizure. When we think about now how we would apply something like this to an X-linked gene, which again is a major advantage over base editing or DNA editing strategies, where we can target a promoter perhaps of one of these inactive genes, in this case CDKL5, that would sit on the inactive X chromosome here and is silenced via DNA methylation. So this is indicative of this gene is not being expressed. The question is, can we drive this inactive CDKL5 that sits in the silenced X chromosome in a very specific gene-targeted manner and overactivate it or reactivate it, I guess, from the inactive X chromosome to compensate for the active mutant allele? In 2020, we actually published a paper on this where we demonstrated that when we target the CDKL5 promoter with these CRISPR tools, so the DCAS9 DP64, so this transcriptional activator fused to DCAS9 that I introduced earlier, or this TET1 enzyme, which is another one of these epigenetic eraser enzymes that can actually remove DNA methylation, we were able to demonstrate that this gene can be reactivated. And we termed this an artificial escape based on the fact that actually, despite being on the inactive X chromosome, a whole bunch of genes are being transcribed still from the X chromosome. These are called escape genes. So our hypothesis here was, can we actually model the chromatin signatures of what one of these escape genes is? What we find in this paper, amongst other things, is that targeting the CDKL5 promoter results in DNA demethylation. So these graphs here, where a combination of DP64 and TET1, or only the TET1, can significantly demethylate the promoter. We can also remodel chromatin, so this H3K27 trimethyl, that stands for histone 3 lysine 27 trimethyl. So that's one of these amino terminal tails from the histones. We can also kick that off too. So that means that we're actually remodeling chromatin directly of the inactive X chromosome to maybe resemble more of what the active X chromosome is. Now, from a translation perspective, this is really, really critical though, where the question is now, cool, this was all done in cell culture, this was actually done in an immortalized cancer cell line, and the question is, that doesn't have anything to do really with CDKL5 deficiency disorder. To be fair, it was a neuroblastoma cell line, so it has a little bit of neurodevelopment, but the only reason we chose this cell line is because it actually has a polymorphism in the CDKL5 gene that was able for us to trace back what the inactive allele is and what the active allele is. But again, doesn't have anything really to do with the disease itself. So what we're really focused in on the lab, other than showing now in this case, look, there's proof of principle that we can do such thing, and we're actually the first to show that you can reactivate a gene on the inactive X chromosome in a targeted fashion, which is as a scientist, is a pretty big deal, and then obviously also has a big impact possibly on other X-linked intellectual disabilities. But the question is, how do you translate something like that into the clinic? And I already mentioned to you before that AAVs, specifically as a delivery modality, have one major issue, which is the size of the cargo is very limited. So DCAS9, where you fuse a DP64 activator and one of these TET enzymes for DNA remodeling on, rapidly exceeds this 4.8 kilobase size limit. But I also introduced earlier already this idea of this modular system in which we broke up the DCAS9 into two halves. The idea here now was, can we just do the same approach here, but then rather than just having a single AAV that's oversized, can we just package two AAVs? And this is a Indian transsplicing mechanism where this N-terminal half finds the C-terminal half once it's transduced in the cell and forms a full-length DCAS9 protein with the activators. And there's a preprint out, by the way, that I just really quickly want to highlight. So if anybody's interested in this more, we're in the process right now in revision to hopefully get this accepted. This work was funded through the Lula Foundation. I'll get back to that in a minute, but this is the #1 CDKL5 patient foundation, as well as CIRM, the California Institute for Regenerative Medicine. They gave us about $2 million award to study this and really translate these epigenetic CRISPR tools into the clinic. Now in the lab, we work with a lot of in vitro models, so cell lines, but we also work with mouse models of human disease. So this is a mouse model now in this case of CDKL5 deficiency disorder. And this is a female mouse that now has one defective copy of CDKL5. And usually what we have to do in these experiments is compare it to a wild-type line. So what hopefully becomes very obvious from already these brain tissue sections, where the wild-type level has a lot more positive cells for CDKL5, which are these brown strings that you can see here, than the center panel. The center panel is a heterozygous female mouse that has a defective CDKL5 copy. Now, when we introduce our epigenetic editor in form of an AAV via an ICV injection, so this is a direct injection into the ventricles of the mouse brain, we actually identify that this DAB labeling for CDKL5 increases, so we get more CDKL5 positive cells, indicative of that we're actually reactivating CDKL5 in a mouse brain, which is also represented and quantified both for the cortex, which is this red area here, as well as for the hippocampus over here, quantified in the bar graphs here. What you can see in the green dots here is that the CDKL5 expression is almost, statistically, is indistinguishable from what the wild type levels are in both the cortex as well as the hippocampus. In science, it's really important to do orthologous or redundant assays, so we also did a Western blot again for CDKL5 expression, in which we actually just grabbed total protein lysate from the mouse brains, run a Western on those, and what we see in a similar fashion, albeit there's still a significant difference between the wild type and the treated group, but we can significantly increase CDKL5 protein levels in the mouse brain. CDKL5 is a kinase, so it has phosphorylation targets. One of the other things to show is what you're actually turning back on, is that functional? Does it have an effect? And what we're actually finding for this phospho-EB2, the function of EB2, what it actually does is not super critical right now, but basically what we're finding is that we can rephosphorylate EB2 in the CDKL5 treated animals, both again by Western blot, but also by immunostaining. So it's the same idea, wild type on the top. In the diseased mouse, there's no expression whatsoever of the phospho-EB2, and then once we treat it with the transcription activator and the TET1 remodeling enzyme, we can turn it back on. Mice are also really interesting because you can actually do specific assays on them that maybe prove things like motor dysfunction, but you can also look at behavioral and cognitive phenotypes. What we did with these mice is we actually put them into this open field assay that proves that mice that have CDKL5 deficiency disorder, as much as you can say it, that are models for this disorder, have a hyperactive phenotype. Again, keeping in mind it's a mouse. Mouse brain is very, very different from a human brain. There's a lot of evolution that has happened between mice and human. So mice really don't get CDKL5 deficiency disorder, but we can recapitulate some of the circuit dysfunction in these assays. And what we find here again is that our treatment reduces this hyperactivity phenotype in the open field assay, and we also tested for the spontaneous alternations in the Y maze. And similarly again to the open field assay, the phenotype observed in these mice is normalized to wild type levels. These are all very critical things to then move forward with to things like the FDA to show, look, we actually have proof of principle, proof of concept data to show that these are actually effective. We're also working on some safety data to move this forward, perhaps to either a larger animal or at the very least to go into some kind of early clinical trial testing. But I'm not a huge fan of working with mice, and part of there's the ethic concerns that come with it, but also I just don't really enjoy going into the vivarium. We have a showering facility, you need to put on a bunny suit. It's a lot of work. Kyle does all the surgeries, he's fantastic at it. I'm scared of touching the mice, to be honest with you, so I'd rather work with cells. So, and one way for us to do this, again, is what I introduced earlier already, is that we have these patient iPSCs. So these are directly derived from a patient, where we can do a blood draw, for instance, which is what the really neat part about these X-linked intellectual disabilities is that when you draw blood from a patient, you might end up with cells that have the mutant allele on, and you might end up with cells that have the wild type allele on, again, because of this random mosaicism that occurs during X chromosome inactivation. What it allows you to do is you actually get an inherent control that is healthy versus the mutant line. We can then actually take these iPSCs, turn them into either neural stem cells, which is a progenitor state. They can then be differentiated into 2D neurons, or we can get these iPSCs, turn them into NSCs, neural stem cells, and then turn them into cortical organoids. You might have heard of these as mini brains. It's really not that sophisticated, but again, the idea is these are actually forming circuits, neural circuits. You can actually measure electrical activity, and what I'm showing you here right now is we just probed for the expression of specific excitatory markers that you would normally find in the human brain, inhibitory markers that you find in a human brain, astrocytic markers, so support cells of the brain. And what we find is that, yeah, these organoids, clearly they're not a human brain, but they express a whole bunch of markers that seem to be very reminiscent of very early human neurodevelopment. Which again is nice because now we can actually test our therapeutics directly in the human background. That is also a 3D shape, right? It's not just a 2D layer of cells, but you actually have an interactome of cells. So cells in a 3D structure. When we target these organoids now with our epigenetic editors, what we find here in this left plot is this is a volcano plot, just showing expression of all these genes that are deregulated in the cortical organoids in the mutant compared to the wild-type cells. What we're able to do actually from the top panel to the bottom panel is we are able to normalize CDKL5 downregulation. So again, it will be down because it is actually loss of function mutation in these cells, and that means that the mRNA gets degraded. And once we actually target this with our activator domains, we're able to no longer have CDKL5 being a differentially expressed gene, so it loses significance, indicative of that it's being normalized. We then also plugged in all the differentially expressed genes into this heat map that you can see here. The nice thing is that these CDKL5 deficient organoids actually have a phenotype that shows that there's a transcriptional dysregulation. Treating these with our epigenetic editors normalizes the state of many, many different genes, more so similar or no longer significantly different from the actual control cell lines. So there's this vast transcriptional reprogramming that happens once we target this with our Cas9 CRISPR modalities. And here's another little plot. You can then, because RNA sequencing gives you so much information, you can grab all these different genes that are differentially expressed and plug them into some kind of matrix. As humans, we like to see patterns and we like to make sense out of things. This is one way to do it. We say, can these genes actually group into different categories of gene expression profiles? So this would be a gene ontology analysis. And we find terms that actually make a whole lot of sense for CDKL5 deficiency disorder to be rescued, such as kinase activity. Again, CDKL5 is a kinase, but we also find very neurospecific terms to be normalized. As a last little neat thing about these organoids is we can also plug them onto these HDNIA plates, which are multi-electrode arrays that can measure 26,000 different cell interactions with each other on an electrical level. So what you can see on the panel here is the control versus the CDD ones. In CDD, you can see a reduced firing rate across these cortical organoids. So that means they are actually electrically, there's a deficit, something is wrong with these. And when we introduce our epigenetic editor, it doesn't look quite like the wild-type lines, but at least we are turning back on the network activity, and we might actually be overshooting it a little bit. So maybe this has a little bit to do with that you can't fix quite everything, but at the very least we are bringing back some activity. I'm not an electrophysiologist though, and I'm very grateful for our collaborators and the people that actually do these assays. And then as a very last example that I want to bring up is the idea of CRISPR silencing for dominant disorders. So this is again another topic switch. And this is probably the most unique situation, or the best case scenario, where really, again, you don't have the option to actually introduce a gene therapy vector here, but rather you go in now in a dominant disorder. An example of that is Huntington's disease, that is actually caused by a CAG, a triplet expansion in the Huntington gene, that causes a neurodegenerative disorder. We published a paper on this also earlier this year, demonstrating the opposite now. Rather than removing DNA methylation from a gene promoter, we use this Cas9 CRISPR modality now to deposit DNA methylation, where we actually silence the gene of interest. So again, we want to bring down the mutant allele, or the mutant expression of Huntington. And we're able to do this in a durable fashion. So this is another advantage of epigenetics. Anybody here that is familiar with epigenetics, really the concept of epigenetics is that these are mitodically and to some degree also meiotically stable marks. So they get transmitted mitodically from cell to cell. So theoretically you have to hit a cell once with an epigenetic mark that then gets stably transmitted to its daughter cells. So from a clinical translation perspective, that will mean that in an approach like this, theoretically, you would have to do a single administration of an epigenetic editor via, for instance, an LNP, a lipid nanoparticle, that then goes away, so you wouldn't even need the constant exposure of the AAV that deposits the silencing mark and then goes away. This paper is very cool, and it shows that we can actually do this durably over several weeks. So theoretically, again, in a treatment paradigm, that would mean that you would have to administer perhaps a drug like this every month. So just to summarize what we do in the lab, basically it's all centered around neurodevelopmental and to some degree neurodegenerative disorders. In this case, I showed you the idea of, I introduced epigenome editing to you, in which we have now really cool tools available to us. So these CRISPR technologies, again, they can be used as gene scissors, can be used for base editing, prime editing, which are all really, really cool technologies that are evolving and developing constantly as we go. New CRISPR variants come out on the weekly. Where we are really focused on right now are these epigenome editing strategies. Again, with the major advantage, it's sort of a one-fits-all approach, where all you need to have available for you is a targetable gene promoter. If you can do that, you can theoretically rescue many, many different genetic mutations. We're testing these in animal models of disease, and again, in my favorite here, these patient-derived cell models, and then delivering them to the brain via adeno-associated viral vectors. This is an overview of all the different most of the different projects that are ongoing in the lab, and sort of where they are. So you see different indications. For instance, Angelman syndrome is an example of another one of these developmental epileptic encephalopathies. CDKL5 deficiency disorder, Rett syndrome. So we have a whole project on extinct intellectual disabilities going on. Specifically, again, around this idea of epigenetic reactivation strategies. We have approaches for autosomal dominant disorders, Huntington's disease, Jordan syndrome, and some other various activities that are all in different stages. CDKL5 is our trailblazer. Whenever we go to talk to other patient foundations, they ask us, how's it going for CDKL5? Because they're trying to sort of hop on that train, right? And the idea is, again, with these approaches, we're trying to de-risk a lot of technology for the next generation then of diseases to come through. With that being said, I want to say thank you. First of all, obviously, thank you all for your attention, and I want to give a huge shout out to the rare disease community. So with the research we're doing, there's a massive over-reliance on patient foundation funding. It's really tricky. It's already been challenging to get NIH funding. It's not getting easy, unfortunately, as we go on. So again, this probably needs to change, but we're trying to come up with smarter ways to approach this. I mentioned that we have platform technologies that are being developed. So theoretically, you can envision, hey, let's group all the haploinsufficient genes into one category and see how well these respond, for instance, to CRISPR activation strategies. Let's look at all these extinct intellectual disability genes together and have these maybe basket trials where all we have to do theoretically is switch out a guide RNA that can target a different gene promoter and maybe make these things a little bit more cost effective. With that being said, being in California, that's really, really nice because we also have access to CIRM. So this is the California Institute for Regenerative Medicine. They were renewed in 2020 for $5.5 billion. They're up on the ballot again. If you think this is probably going to be up on the ballot again at some point in 2028 or so, if you think this is cool science, things like that is generated obviously through research funds that are really, really critical. So just be on the lookout up for it. And again, if you have any other questions beyond this, grad school or what it's like to be a scientist in the lab, I also put down my email and Kyle's email, and I'm happy to take any questions on email or right now also. Thank you so much. I'm about to take a picture of everybody here, so I told my wife last, hey, can I have a picture?
Everybody smile.
Thank you so much, everybody. Sorry about that. Are there any questions?
We're going to do questions in just a moment. But what we're going to do is take about a 5-minute break. So if you have questions, please stick around. If you want to come see me for a clipboard, come down. We're going to make some 2 lines here going out the door. But we'll resume questions in just a moment.
You guys don't like check in?
Oh, we're going out. out in the lobby. Doc David and I are going out in the lobby.
What the genetics are, what the genes do, and then how confident are we that if we reverse the gene
All right, let's start questions. And so, if anybody does have questions, raise their hand, and we're going to pass around this.
When you guys are talking about using the ADPs for the haploinsufficiency, what have you seen or what have you tested to prevent overactivation that you were talking about for like genes you didn't want to be activated?
So, in haploinsufficiencies or like those Goldilocks indications with AAV, how do you prevent the overexpression?
Overexpression of genes you don't want to be expressed.
Yeah, so I think like one of the beauties of what Julian explained with like the CRISPR system, and again in the Function 1 paper that we published earlier this year, is that we can really do like almost what we call, and I think the title of the paper is like tunable gene expression, right? So in essence is by the combinations of effector domains and different things with CRISPR, is you can turn up, you can take a gene from 1x to 1.5 to 1.7 to 2 to 4 or whatever. And so we can really kind of selectively pick out the combinations of effector domains. If you're looking at like strict gene therapy, so the example that I gave, the 2 trials that are ongoing with AAV for Rett syndrome, is both of them have used different systems that prevent the promoter of that AAV to be overexpressed. And so basically they're delivering a promoter that has brakes on it. And they went through, both of them are different approaches. And so basically the more gene that's being produced, the more active these suppressive mechanisms are. So even if you give 1,000 copies of that AAV to a cell, is the MECP2 level will never exceed 2x. You want to add anything?
No. I guess the biggest thing with these haploinsufficiencies is they're caused by loss of function. So really, even if you were to overactivate at least a mutant copy, because that's often the question that we're getting is what happens if you get a mutant allele, does it have an effect? At the very least, we can say that from that level, it really shouldn't have an effect on the cell health. Obviously, if you then go for the wild type level and you boost that through the roof, that might be very tricky. And then the idea of off-target effects that you maybe get with CRISPR, beyond the actually intended editing side, that's a whole other beast that I think you just kind of have to empirically validate to some degree, but it's also, it's a little bit of a, you see what you look for basically, and I think often we just have to be very honest about what actually is an off-target and how has it been reported. So sequencing technology and all those things allow us to really go into depth, but often that again comes also with maybe increased cost of the actual experiments and things like that. Yeah.
During your guys' presentation, you guys mentioned a viral vector and also lipid nanoparticles, and I was curious, like, are those 2 different ways to deliver treatment to patients with rare diseases?
Yeah, that's really cool that you picked that up. And so, as Julian mentioned, so a lot of times when we think about CRISPR technologies, other types of technologies, we kind of think of what the cargo is and then what the delivery modality is, right? So we can build really cool CRISPR systems, and it works really well in vitro in a cell in a dish, and then we have to figure out how do we translate that, how do we get to the tissue and the cell type that's the most needed. Right now, things like lipid nanoparticles are great, and like this story of baby KJ was a lipid nanoparticle that's delivered because that specifically affects the liver, and everything likes to go to the liver. Right now, with lipid nanoparticles, is still in its very infant stages of demonstrating that it can work in the brain. And so right now, there's not lipid nanoparticles that we know readily cross the blood-brain barrier, distribute well, and hit a large number of neurons. But what we do know is that certain types of AAV therapeutics, when delivered either in young patients or delivered directly into the cerebral spinal fluid, can transduce a large number of neurons in the brain. And so right now, as Julian mentioned, AAV is kind of the gold standard for how do you treat the brain? How do you get genetic information into the brain? And that genetic information could be the coding region of a gene, like Julian mentioned for like haploinsufficiencies. Could we just deliver this? The example of spinal muscular atrophy Can we just deliver the coding region of that gene back under any promoter of choosing? Or can we deliver genetic cargo such as CRISPR epigenetic editors to regulate gene expression or turn back on the already existing copy of a gene within a cell? And so right now, AAV's kind of the gold standard. It's not perfect. There are concerns, safety concerns, dosing concerns, efficacy concerns with AAV. Also, we didn't talk about AAVs are becoming incredibly expensive on a per patient once they get approved. But a lot of the things that Julian and I think are focused on is how do we optimize AAV therapeutics? How do we look at AAV biology to create better therapeutics to access the brain better while we're waiting on some of these maybe second, third, fourth generation type technologies, synthetic biology, LNPs, polymer nanoparticles, viral-like particles, to catch up to how good AAVs are for the brain right now.
Yeah, that's probably not a sustainable model for health insurance. But it saves your kid's life. I guess it's one of those strange balance.
During your presentation, you discussed using different kinds of cells, and most of your research was on eukaryotic cells. Have you guys done any work with prokaryotic cells, like experimenting with different bacteria that make different kinds of antibiotics and such, and editing those?
Yes, we work with them almost on a daily, but we use them as biofactories to make our plasmids. So really we use specific modified E. coli strands all the time that we can just easily grow up, that have some antibiotic resistance gene. Then we can extract for instance, our Cas9 plasmids from those and produce AAV from that plasmid. But it's not necessarily prokaryotic biology that we're studying, if that makes sense. But we'd rather just use them as a tool, as a biofactory for the eukaryotic experiments. And then obviously Cas9 being a prokaryotic enzyme, that part is I guess, is a given.
I'm clearly not the smartest person in this room, so please forgive this question. But during your presentation, you spoke about baby KJ and the CPS1 treatment that he received. You referred to it as bespoke, and that he didn't receive it until he was close to 8 months old. Is that because it was the first treatment of its kind? And if you're able to make a bespoke treatment for other children, is that something that can be delivered much earlier?
Yes, and I would, there's a colleague of ours just up the road from here in Berkeley. His name's Fyodor Urnov. He's just received a large amount of money and funding. He led a lot of the off-targets and the statistical modeling of all of these things, as well as really working with industry to accelerate the regulatory process to do this. And so they just got a $20 million grant from Danaher, partnered with Berkeley to try to treat 16 more urea cycle patients. And so baby KJ so far is a one-off biallelic mutation of this gene. And so he's the only one. If another baby was born with the same mutation, theoretically you could use the same therapeutic and go for that one. What they're trying to do right now is use this as a template. Earlier this week in New England Journal of Medicine, our current head of HHS and the FDA just published a new guidelines that they're trying to implement at the FDA, that if you have probable mechanistic rescue, so things like Julian's explaining with CRISPR, things like base editing, if you know what causes the disease and you then fix the disease, the regulatory agency should be much more lenient with you, because you know what you're doing, you know how you're fixing or curing the genetic cause of that disorder. But Fyodor has written volumes of perspective articles on this, of how you could democratize, how you could get these to more patients, how you could do this quicker. And he's been writing about it for as long as I've known him, and longer than that. But now they have some financial backing from industry partners to try to do this much, much quicker. And again, with the idea that you're going from an N of 1 to 16, within the next couple of years, and then hopefully that leads them to doing, potentially hundreds. Again, once they're kind of working through the pipeline, the regulatory, what's the conversations with the FDA, what type of safety data do you need to generate, what efficacy data do you need to generate per mutation per person with a base editor before you launch that clinical trial. Julian and I are trying to do the same thing with not going mutation by mutation, base by base, but saying classes of disorders. So if we know that the gene is low and we can bring it up, if we know the gene is off, but there's a good copy that we can reactivate if the gene is a dominant negative and we can silence that, could we then do that across classes of mutations, across classes of disorders? And so again, lots of the CRISPR folks are all circling this idea of how do we do it, and how do we do it faster? Because the other question we get all the time is, why don't you go faster? So, I think there was one over here. Do you want to add anything to that?
As you guys move into your clinical modeling, is there any specific way that you guys have already thought about designing your clinical models as you're working towards the FDA approval?
Yeah, so we, fingers crossed for all of us, we have a couple of large programs both with the NIH and with CERN right now that are currently being reviewed, or I guess that were reviewed with CERN, and hopefully when the government's now reopened, that will be reviewed at the NIH, where we've given a lot of thought of what are the exact data sets that we need to. So I think as a scientist, and as I would speak to the younger people in the room and scientists, there's multiple steps of, like Julian said, cool science, you show that you can do it, you show that it could work, you can show that it works in a mouse, you can show you could work in a human neuron-like model system within these brain organoids. And so there's certain people that you're gonna have to convince that this is worth moving forward, right? You're gonna have to convince somebody to give you money to fund to do it, and coming up with good clinical trial design, who are we gonna recruit in what are our primary endpoints, what is the safety considerations. You're going to have to convince the regulatory agencies in terms of what model systems do you need to, what is the most appropriate way that would show predictive efficacy or predictive safety toxicity. Which model systems are you going to use to be able to determine the most efficacious dose? The one thing we didn't talk about with AAV is the one and done therapeutic. You get one shot to get the dose right. And if you have an AAV and you are in a gene therapy trial, you're going to be excluded currently now from any other gene therapy trials in the future. So you have to be really, really thoughtful, really, really considerate on that. You're going to have the end users. You have to go to the patient organizations. Is this something that you would be willing to do? Is this something that you could get your community that they would have buy-in to do? And then I think the other one that I will say is because you can probably do all of those things. And then the last one is you have to convince yourself that your data is right. You have to convince yourself that whatever you're doing and whatever you're investing in is appropriately designed, that you picked the right endpoints, that you looked at the right off-targets, that you looked at the right safety, you determine the right dose in a way that you're going to believe that would be safe to move forward and then have a chance to be efficacious. So we've spent lots of time doing this, specifically with the lead indication of CDKL5. We do this with conversations with the patient community, with the patient advocacy groups, and then with clinical experts that see these kids every day. And then what also is realistic to do with things like AAV delivery for the brain, and knowing what those limitations and safety concerns may be.
So since we're on regulation, with current environment, is this getting easier or worse? And I know funding is becoming more and more challenging, but is it getting easier to get to clinical trials or worse?
Supposedly easier for these kind of bespoke things. Again, using the baby KJ example as the front runner. But in reality, it seems more chaotic than ever. So it's hard to see, I guess, the plus. But I think there's some policies in place that are supposed to fast track a lot of these things, including also another example of maybe relevant to this talk, that for instance, the NIH the FDA has a modernization act where they're saying you don't even need mouse models anymore, but just show it to us in a complementary model like these organoids, for instance. Which I think is especially relevant to something like these CRISPR therapeutics where you actually target human DNA sequence. And you would have to build a mouse avatar, basically, a mouse therapeutic in parallel. That's some of the fast tracking. Maybe Kyle has some other thoughts?
So Julian hit it. I just wanted to emphasize again. So Peter Marks, the former head of the FDA, and he put into place some really critical things in motion before he was no longer with the FDA. And a lot of that came from learnings actually from the pandemic and the development of the COVID vaccines and mRNAs within lipid nanoparticles or adenoviruses. And what the FDA learned from that is that the constant communication with researchers and clinical development teams greatly sped the clinical process. And so before, when you would write something to the FDA, you would write a formal letter, document, package. It would go to the FDA, they would receive it, they say, we'll get back to you within a certain amount of time. They're going to review it, they're going to give you very formal documentation back, you're going to get that, you're going to have a certain amount of time to read. And so every cycle that you're going through, hey, we have a question on our clinical protocol that we think that maybe X is slightly better than Y, or we want to alter the dose slightly because of what we learned. Each time that you would do that with the FDA, that turn of the wheel would take months. What Peter Marks started within Operation Warp Speed and the development of the COVID vaccines was just talk to the FDA constantly. Email us when you have a question and we'll get back to you in real time. We'll still do the documentation. We'll make it much, much more dynamic, right? So now the sponsor, the developer, or the PI of the project could go, hey, a lot more transparent. We're not going to hold 10 questions at once because we know we're going to get delayed by a month. We're just going to shoot them off of it as they come. We're going to have those real conversations. We're going to develop this in real time. And that's going to speed the process. Better interact meetings, quicker pre-IND meetings, better information that you're getting from it. And so in theory, all that's still in place, except the staffing at the FDA has been gutted, right? So when you try to talk to them, there's just not people there to reply to the requests coming in. So we hope that comes back to that level. That again, a lot of these policies that were initially implemented, and then I think, in science, certain things happen, and everyone's kind of like blown away. And I remember we were sitting, I think, next to each other when Kieran presented baby KJ's story for the first time, and literally we were getting text messages from rare disease foundations as that was happening, right? And that's really started to change, again, the way that the FDA is thinking about these therapeutics and their guidelines because of the success of one base-edited baby.
So one other question for you. Peripheral myelin protein mutations. Do you know anybody doing work on that, specifically like HNPP or CMT?
I do not, unfortunately. Myelin sheath proteins?
Myelin sheath proteins, yeah. HNPP, hereditary neuropathy with liability to pressure palsy, or CMT, Charcot-Marie Tooth disease. They're both, one's a deletion, one's an insertion.
CMT, I'm
peripheral
familiar.
myelin protein.
Somebody's working on CMT.
Yeah, actually, I just hired a postdoc from France. She researched CMT and I think read-through drugs for that in France. But I am not familiar with it, other than I'm familiar with the disease. And I don't know who here, and maybe somebody is also know.
Fletcher was working on some demyelination disorders.
Perhaps some people at UC Davis.
I just wonder what the delivery system on something like that would be, because that would be quite a bit different than the CNS problem.
The peripheral would probably still use similar viruses, but
your delivery would be different, right?
It'd be a systemic delivery. So like
So systemic delivery instead of a CSF?
So like the example up there, Duchenne's muscular dystrophy. Sorry. The example up there that we had, Duchenne's muscular dystrophy is direct injections like into muscle. And so again, with peripheral nerve, peripheral targeting, again, you could do AAV administrations. It's just going to be systemic, not localized within the CSF. Yeah, I don't know who's working on viral base for demyelinating disorders.
It's okay, I don't need the speaker. I think I'll just speak up, plus I'm not good at catching. The question I had is, I think CRISPR's been around, what, like 10 years, 10 plus years, right? Roughly, more or less. And you guys are experts in the field, so do you guys suspect that there may be some sort of new tool that could be enhancing the research in the field that could just have as big of an impact as CRISPR, or something similar like that?
Yes.
would it be like within the next few years, or do you think like it would take longer?
It's really tricky to say. CRISPR was discovered previously, also before it made the news, basically, right? Because it was already, I think, 5 years before Jennifer Doudna and Fink published their milestone papers. There were actually already manuscripts out describing these CRISPR viral defense mechanisms. I think it just comes down to biotechnology, mining microorganisms, mining different species, understanding the mechanisms. But it all, again, also comes down, I think, to funding these very basic research efforts that then result in these very, very impactful, life-changing technologies. But I'm confident that there will be something that is even better, even faster in our lifetimes, or sooner than later, actually.
And so a quick follow-up then, if more research labs implement AI to analyze their data and so forth, do you think that would accelerate it much quicker? But I'm not sure how the funding goes with all that.
Yeah, I think AI has a lot of promise and maybe also a lot of hype. I think we'll have to see how that goes. I think it still requires some kind of human user, and somebody to understand what they're actually looking at. And also, I think the idea of these AI hallucinations is also very real, where you just get complete nonsense out of your prompts. I think, but these things are also developing constantly, so I don't want to write it off completely. I think it's a very, very useful tool, especially from the mining perspective. But you can maybe make the argument that we were already able to do it maybe a little bit slower. Again, this will probably contribute, especially when we think about protein engineering, that we can maybe enhance some of these Cas proteins and make them better, maybe reduce off-targets by just feeding these large language models a bunch of data and having some kind of output that says, with a high likelihood, this will be better than what you're using right now. Maybe only incrementally better, but still, I can envision how this will probably make some things better, but I don't think it will be the be-all, end-all. Exactly.
Yeah.
That's my take. I don't know if you have other thoughts on that.
I mean, like my lifetime as a scientist, we've lived through like iPSC discovery. And everyone was like, whoa, like that's the coolest thing that's ever going to be ever discovered of all time. And it's going to cure everything. And then 10 years later, CRISPR technologies, and everyone was like, whoa, like, again, problem solved, we're good to go.
Same with shRNAs too, right?
shRNAs, evolved AAVs for delivery into the brain, like all, there's breakthroughs that happen, I think, in specific fields all the time. These very large revolutionary changes, right? iPSC technology, CRISPR technology, and especially like the ease of use and the cost of developing CRISPR technologies. Like I think those were big breakthroughs, and one happened in 2004, one happened in 2012-ish, right? Then, like, so again, I think those advances are going to happen, those breakthroughs are going to happen, and again, I don't think, with iPSC technology, not everybody was sitting around going, like, wow, I really wish I could reprogram a fibroblast into any cell I want. But then all of a sudden, like, then you see the data, you see the results, you see that, like, massive stride forward. And so again, AI may assist with that, but as Julian said, it's going to be a lot of whatever data you're feeding it and whatever queries, whatever questions, whatever answers you're trying to get back out, right? So, I think if you just ask ChatGPT, hey, what's the next big scientific breakthrough in the vein of CRISPR or iPSC, like, again, I'm guessing it's we should ask it later. Yeah, we got a long drive home. We'll figure it out. We'll let you know. But again, I think, again, it's going to be exactly as Julian said. It's really, really good with condensing and summarizing data. It's really good at mining data and looking at things maybe in new ways that, human eyes haven't quite yet. But it's still going to be dependent on the data that's generated to go into it, right? Like again, there still has to be input to those systems.